Inicié. Ah, ok. Muy bien. We are online now.
I can't see how many people are logged in the Zoom meeting right now, but we can wait a couple more minutes before we can start.
Sure. Can you actually like tell how many people? I guess you could. There's like YouTube, like the video log. It will log how many people watch it, or
sometimes they tell you the number of participants at 18. No, not
gotcha. Gotcha.
The last day, and everyone is thinking on Christmas, so
oh, of course, yeah.
And time off, so
when people are traveling or just getting everything wrapped up, it's a busy season. We're thankfully not traveling this Christmas. I'm so thrilled.
Are your parents coming down to visit you?
Yeah, they have a house in our neighborhood, so they're.
They do. Yes, you mentioned that before. Yeah, I forgot. I thought that they were still in DC.
Right. No, they moved down in 2018.
Yeah, you mentioned that. I forgot.
Yeah. No, it's great. We get to do frequent dinners with them, and the girls are getting to know them well. It's sweet. And
Jesse's parents are also in Houston?
No, they're in Arkansas. So we visited them over the Thanksgiving break in November. So kind of it's like one holiday a year is enough, go up there. We try to do a week in the summer as well. So kind of split it up that way. They have beautiful land in the middle of the Ouachita National Forest, surrounded by trees and mountains, and it's gorgeous. It's very different from urban Houston,
It's probably beautiful there.
Yeah, there's no cell service, so you get to really focus on the important things. True break. I sure wish this was in person. I would love to visit you guys in Mexico City one day.
Yes, well, hopefully next year things will get better, and then we can resume our in-person seminars, and it would be great if you can come down and visit us.
Yeah, that'd be fantastic. We'll keep working hard and get some new stories. We have some new content for you guys.
Perfect. You love it there.
Yeah, I've never been to Mexico City. I've been in northern Mexico a few times, but not in central Mexico.
You have to come down. I promise I'll put something together.
All right.
For you.
Deal. I'll hope you organize it needed.
Okay, so it's about 30 people now in Zoom. I don't know how many on YouTube, but we can wait for 3 more minutes.
Sure. Shut off Outlook so it's not constantly dinging throughout the presentation.
Would you rather hold all the questions until the end, or is it okay if people can?
I always think that's easier just because, so often people will ask a question and
Something that you will
coming up in the
explain.
next slide,
Mhm.
So, I mean, if somebody just cannot wait, that's fine. I love enthusiasm, but it's always easier to answer them at the end.
Yeah, and less interruptions also make
Yeah.
a better presentation.
Exactly. We
Okay,
did a
so-
joint, sorry.
No.
I was just saying we did a joint lab meeting with Diego Bojorquez's lab last week, and their lab had so many questions. It took 2 hours rather than 1 hour to actually make through the
Yes.
presentation. So,
Like, can I get to the next slide, please?
yeah, exactly. All right, thanks.
So it's 12:05. Let's start. Welcome everybody to the last talk of our seminar series for 2021. If we got a silver lining of the virtual seminars is that this year we had the opportunity to host amazing scientists from all over that delivered really interesting talks, and today's lecture by Dr. Buffington will be no exception. So it is my pleasure to introduce Shelley Buffington, who is an assistant professor at the Department of Neuroscience, Cell Biology, and Anatomy of the University of Texas Medical Branch. And this year, she also joined the Seeley Center for Microbiome Research at UTMB. Shelley graduated summa cum laude from the University of Arkansas in 2007 in biophysical chemistry with a minor emphasis in cellular and molecular biology. Then she got her PhD at Baylor College of Medicine in 2012, working with Matt Rasband on the structural plasticity and molecular diversity of the axon initial segment. Shelley then joined Mauro Costa-Mattioli's group for her postdoctoral training at Baylor College of Medicine, where she worked on the mechanisms by which gut microbiota influence neurodevelopment, plasticity, and behavior, and the translational control pathways regulating synaptic plasticity and learning and memory. She has authored several papers in the most prestigious journals in the field, including Cell, Nature Neuroscience, Neuron, and Human Molecular Genetics. She and Mauro Costa also hold a patent for probiotic therapies for developmental disorders, which claims a new method reversing social deficits in a subject suffering from developmental disorder, including autism spectrum disorder, in comprising administering a specific bacterial strain. Over the past decade, we have seen an increasing interest in the role of microbiota as a previously unsuspected regulator of brain function by making up the denominated Guttobrain axis. This axis is gaining ever more traction in fields investigating the biological and physiological basis of psychiatric, neurodevelopmental, age-related, and neurodegenerative disorders. The work performed by Dr. Shelley Buffington has contributed to our understanding of how particular bacterial species can influence behavior. So once again, thank you, Shelley, so much for accepting to share your work with our community. And without any further ado, here's Dr. Shelley Buffington.
Oh, Luis, thank you so much for the invitation. I'm really pleased to be here, and thank you all for joining the talk today. I'm going to share my screen. Let's see. Yes. All right. I think that should be good. You guys can see everything and hear me?
Yes.
Okay, excellent. Yeah, so as Luis said, I made a big change. I mean, it was still in neuroscience, but going from basically the cell biology of neurons to really more how the body as an integrated system and how gut microbiota can affect brain development, function, and behavior as in my postdoc. So I'm going to tell you mostly stories that came out of that postdoctoral period. Of course, that's inspiring the current work in our lab. And so I'll give you a little insight into that at the very end of the talk. All right, so just to give you a very brief overview of the gut microbiome, I'm sure many of you are familiar with it, but I want to just provide some context for how these seemingly, small and maybe insignificant fellow travelers of ours in life can actually have a very outsized impact on our even brain and behavior. So what is the microbiome? Well, it's of course the collective term of genes that are expressed by microorganisms, of course, bacteria, viruses, and fungi. Primarily today, we'll focus on bacteria. There are roughly 10 to the 13th bacteria on and inside of our bodies. And so that actually roughly outnumbers our cells at about a 1.3 to 1 ratio. And of course, there are 500 to 1,000 bacterial species occupying our bodies at any one time. And of course, given that each bacterial strain has a genome that contains thousands of genes, that can greatly expand upon the human repertoire available to us, and actually provides for some very beneficial physiological functions. And of course, there's also pathogens that are associated with disease. And finally, just characterization of the gut microbiome and the associated metabolome has been made possible and also accelerated the technological advancement in high throughput sequencing techniques over the past 2 decades. And I think one reason that this field is actually really burgeoning right now is that the price of sequencing has come down so much that it's much more accessible to labs. And so it's a really exciting time in the field where there's lots of new discoveries being made. So it's wonderful to be working in this field. So how do we know the microbiome can contribute to brain function and behavior? Well, there's been a lot of great pioneering work, from Borsheli's Diaz Heights and Sven Petersen at the Karolinska, as well as out of John Cryan's group in germ-free mice, showing that microbiota are essential for the development of neurotypical anxiety-like and even social behavior. So in these graphs in A, what you're seeing is an adaptation of the Crawley's 3-chamber test for sociability. And what you see here in the middle is a subject mouse, and it's got the opportunity to either interact with the mouse in this left chamber or an empty cup in the right chamber. And the investigators measure how much time the mouse spends interacting with the other mouse versus the empty chamber. And what you can see, at least in this conventional colonized cohort, there's a little bit of preference, but not significant for the mouse. But what you can see here in the germ-free cohort, so these are mice that are reared in the complete absence of any microbiota, there's a strong preference for the empty cup. And this is has often been recognized as social dysfunction or even autism-like social dysfunction. But what you can see is once these mice are colonized with just gut microbiota via fecal microbiota transplants from conventionally colonized mice, that restores a clear social preference for the mouse over the empty cup in the animals. Now, more recent work out of Elaine Chao's lab at UCLA has been employing more advanced techniques, including next generation sequencing, modern imaging, and metabolomics. And then they have recently revealed in a Nature paper from 2020 that the maternal gut microbiome generates essential neuroactive metabolites that are present in maternal circulation and in the fetal brain of offspring that do influence behaviorally relevant anatomical wiring of the brain. And for example, here, this is all looking at thalamocortical axogenesis. And on the left in panel A, what you see is the RNA sequencing results showing that in comparison to specific pathogen-free mice, mother mice that were treated with antibiotics during pregnancy, and these are in the, I think these are in the offspring brain, you see, differential gene expression. And one of these genes that they focus on in this paper is the netrin G1, which is essential for axon pathfinding. What you can see in the offspring brain from mothers treated with antibiotics during pregnancy is you see a decreased thalamocortical axon projections, and that is behaviorally relevant. It induces some sensory deficits in these antibiotics mice. But once they restored this 4 microbial metabolite cocktail, they're able to restore these thalamocortical projections and rescue the behavioral deficits they observed in the mice. And so that's one thing that we're really focusing in my lab is the contribution of the maternal gut microbiome to fetal neurodevelopment and the long-term health outcomes in the offspring. So another important component when we're considering how the gut microbiota impacts the brain, of course, are the communication pathways by which gut microbiota can actually influence the brain. And there are multiple lines of evidence that suggest roles for these microbially derived neuroactive metabolites, like seen in Elaine's lab. And these are released into circulation and are important for the regulation of nervous system development and function. Some of these are even neurotransmitters or neurotransmitter precursors. We also know that microbiota-mediated modulation of host immune function is critical to the nervous system. Obviously, there's a critical role for microbiota in immune maturation, but also we know that the immune system is critical to brain function. So microbiota contribute to maturation of the peripheral immune system and to also microglia in the brain. Finally, there are exciting new studies that are literally beginning to shed light on the precise gut-to-brain neural circuits that mediate sensory transduction and reward perception. And this is namely via regulating direct synaptic input from enteroendocrine cells onto vagal nerve efferents. And a lot of this work is done in Diego Bojorquez's lab at Duke and also Daniel Maceda's up at the Rockefeller. So of course, on the other hand, a healthy gut microbiome is critical for normal, healthy neurodevelopment, but we also know that a dysbiotic gut microbiome or a disruption of the community ecology of the gut microbiome of an individual can contribute to host disease phenotypes. And of course, we know that this is true in several neurodegenerative disorders, including Parkinson's disease. For example, this is a somewhat recent paper showing that injection of exogenous prefibrillary tangles into the gut can actually be translated upon the vagus nerve to the brain. Upon vagotomy, those are no longer able to travel up to the brain. So this is a subdiaphragmatic bilateral vagotomy. And so they can block this, of course, with the SNCA knockout mice with no Parkinsonian development. Also, work from Aaron Elenov's lab at the Weizmann has shown that gut microbiota can make critical contributions to demyelinating disorders. And this is kind of the opposite end of the spectrum, where in these SOD1 mice, which are mouse models, a genetic model for amyotrophic lateral sclerosis, when the mice are treated with antibiotics, this exacerbates disease onset and leading to a very advanced disease within just 140 days. And so clearly microbiota are making essential contributions to the host immune system and others to prevent autoimmune disorders. And then finally, it's also been shown that the gut microbiome can contribute to epilepsy. Even it's been shown that a ketogenic diet can help dampen epilepsy phenotypes. This is again through metabolites. So we know that dysbiosis is this disruption in microbial community, and how do we study that? And so often in either mouse models for human disease or even in humans, what we do is we perform 16S gene amplicon sequencing to identify the bacteria that are present, identify the composition, and we can also do either predictive metabolomics from that data or even real true metabolomics. We can identify clustering of the healthy gut microbiome that is disparate from those individuals with a disorder. So for example, either those individuals with the disorder can be lacking a certain gut microbiota that promotes health, or they can be added with a opportunistic pathogen, for example. So here, the work that I'm going to tell you about today is primarily focused on neurodevelopmental disorders, and many of the mouse models are for autism spectrum disorder. So of course, we know that autism is has a strong heterogeneity. It does have a strong heterogeneity, but also a strong heritability. And so we know it's primarily based on genetics. However, there's growing consensus behind this idea of a two-hit model where environmental factors, including those that are listed on the right side of this slide, can actually tip the balance of somebody that's genetically predisposed to autism toward phenotype manifestation. And the data that I'm going to tell you about today is primarily focusing on the effects of maternal diet. So in part one of this talk, I'll tell you about what yielded the data in this Cell paper from 2016 and where that got started. So we know that worldwide, there's been a significant increase in the incidence of obesity. And this is true not only among the general population, but also among women of childbearing age. And furthermore, it's not only been in the US and other more industrialized countries, but also like it's growing in areas of Africa as well as over in China. So this is really a global problem. And I like to say that this is actually maternal diet-induced obesity is a major mental health concern, because it does increase the likelihood that the children born to women of obese status will be diagnosed with a neurodevelopmental disorder. So what did we do? So we decided to model diet-induced obesity in mice. And the way that we did this was that female mice were fed either a regular or high-fat diet over 8 weeks prior to mating. We mated the dams Roughly 3 to 4 weeks later, because we did have some spontaneous abortion in our high-fat diet-fed animals, the subject offspring were born, and very importantly, at weaning at 3 weeks, all of the offspring were fed a regular diet from there on. So the only exposure to high-fat diet was during gestation and lactation. And we perform multiple tests for social behavior. One of these is called reciprocal social interaction. It's very simple. You take 2 mice that have never encountered each other that are sex and age matched and cohort matched as well. And you put them in a neutral arena and you allow them to interact. And what you can see is we saw profound social dysfunction in our maternal high-fat diet offspring. Now, all of the data from this paper is going to be in male offspring. And so these maternal high-fat diet offspring much preferred to explore the perimeter and had very brief, if at all, interactions with their interaction partner during the 10-minute test. We also use Crawley's 3-chambered test for sociability and preference for social novelty. This is like the picture from John Cryan's lab and the germ-free mice that I showed you earlier. There's this 10-minute habituation phase where the subject mouse gets familiar with the apparatus. He's allowed to go between all of these chambers. So we can get nice locomotor data that way. In the sociability phase, we place a mouse in a cup, and we place an identical but empty metal cup on the other side of the cage, and we allow the subject mouse to either interact with the mouse or this empty inanimate object. And finally, in the third and final phase, and these are all just back to back, we leave mouse 1 where he is, but introduce a new mouse 2, as a novel interaction partner, and measure the amount of time this mouse spends interacting with mouse 1 versus mouse 2. And what you can see here is neurotypical mice in our hands strongly prefer interaction with the mouse over the empty cup, as well as in the social novelty phase with the new mouse versus the old mouse. However, just like in the reciprocal social task, we saw profound social dysfunction in our maternal high-fat diet offspring. We actually did a third test that wasn't included in the paper, and this is called social condition place preferences. Will come up again later in this talk. And it was really championed by like Rob Malenka's lab has done a bunch with it. Stanford, Gould-Dolan there, who's now at Johns Hopkins, have done a lot with this. And so the idea behind this cage is it's an adaptation of the classical condition place preference that is used in the neurobiology of addiction field. And so what we do here is we take a mouse from his home cage, introduce them into 2/3 of that Crawley's 3-chamber test, and put bedding A on one side and bedding B. Now these A and B are very different from each other. They've got different textures. They've got different sensory, like olfactory cues. And so this is just a 30-minute preconditioning phase. We measure how much time they spend on bedding A versus bedding B. It's all been optimized for kind of equal preference. And then there's 48 hours of conditioning. First, we put the animal on social bedding back into his home cage. This is called social bedding A, and we do counterbalance these, which one's the social versus isolated bedding. And then after 24 hours with his cage mates on this bedding A, we put him on bedding B, but he's isolated. And then 24 hours later, we return him to the apparatus to do a post-conditioning test, and we ask which side of the box do you prefer? Which bedding do you prefer? Neurotypical mice have a significant preference that's developed for the socialized social condition bedding. But again, our maternal high-fat diet mice established no such preference. So clearly we had a profound social dysfunction phenotype in these maternal high-fat diet offspring, but we didn't know what necessarily at that time, what could be driving this. However, we know that diet is a primary driver of microbiome composition. And there's also some historicity driven by early life exposure. So we wondered if the diet-induced changes in the maternal gut microbiome could also be passed down to these maternal high-fat diet offspring. We began looking into this. And here's kind of the rationale that we were thinking when we were designing our experiments. We knew that diet dominated genetics in determining microbiome composition. And furthermore, the maternal gut microbiome is vertically transmitted from mothers to infants. Now, of course, there's a progressive maturation of the gut microbiome that takes place during early development, and this does overlap critical periods in brain development. We also know that obesogenic diets can significantly reduce overall microbial diversity, which is often associated with disease phenotypes and the underlying pathophysiology. So we really sought to determine the effect of this maternal high-fat diet intake on the offspring gut microbiome and whether or not that could be causally related to the social changes that we were observing in the males. All right. So what we did was we performed 16S ribosomal RNA gene amplicon sequencing. Now, each one of these dots that you see here in this principal coordinate cell analysis represents an individual subject. And you can see a clear clustering based just on the maternal diet. Now, importantly, these mice were actually 6 months old, so they had been apart from their mothers for over 5 months, and they still had the sustained dysbiosis of the gut microbiome. It was actually characterized by a significant reduction in overall diversity, as you can see in this graph, bar graph on the right. And so there was a negative selection of species driven not by the maternal high-fat diet and the offspring that led to an enduring dysbiosis. Now, of course, mice are coprophagic, and so this means that they eat the fecal microbiota that's on the bottom of the cage floor. And this is actually an effective means of microbiome transplant between cage mates. And so we took advantage of this fact to begin to test whether there was a correlation between the gut microbiome composition and the performance of these mice in the social behavior tests. And what you can see here is the co-house maternal high-fat diet mice had a significant shift back toward the normal gut microbiome composition, as you can see here, like that of their cage mates. And the mice in this graph were housed at a 3-to-1 ratio of normal to maternal high-fat diet mice, but we actually also did this at a 1-to-3 ratio. And one maternal regular diet mouse was sufficient to rescue the gut microbiome composition of the remaining 3 maternal high-fat diet mice in this cage. And not only did it rescue the gut microbiome composition, but we saw a clear preference develop for the mouse over the empty cup, as well as rescues in reciprocal social and the social novelty tests. So together with data in germ-free mice that I don't have in this slide deck, what this suggested to us is that something important for normal social behavior was missing from the gut microbiome of these maternal high-fat diet offspring. And so we returned to sequencing. And at this point, we performed whole genome shotgun sequencing to see down to the species and even strain level what was present in the gut microbiome of our maternal high-fat diet offspring versus controls. And among the bacteria that were lost or actually significantly downregulated in our maternal high-fat diet offspring, the highest was Lactobacillus reuteri. And actually this graph needs to be updated because just last year, this Lactobacillus reuteri was reclassified to Lamasi Lactobacillus reuteri within its own clade. So anyway, so what we did was we wondered, okay, how could this, could this bacteria be relevant to the social dysfunction that we see? And actually, when we looked into the literature about what was known about Lamasi Lactobacillus reuteri, there was a group up at MIT led by Susan Erdman, and they had found that this bacteria was able to actually expedite wound healing from a skin biopsy test, but that it was oxytocin dependent. And that was really intriguing to us because, of course, like Larry Young's work and that of Tom Enzel has suggested that the oxytocinergic system is absolutely essential to normal mammalian social behavior, even affiliative social behavior. So what we did was we took Lamasi Lactobacillus reuteri, we put it in the drinking water of the animals. We changed out the drinking water every day over a period from weaning to 4 weeks later, so 7-week-old mice in the juvenile period, at which point we did our behavior. And we kept the mice on the L. reuteri throughout the behavior. And what you can see here, just kind of in a summary graph, is that L. reuteri was able to rescue the social behavior of our maternal high-fat diet offspring, which is completely surprising to us. And we thought it was remarkable. We thought at some point we'd have to maybe do a cocktail of these bacteria, but Lamasi Lactobacillus reuteri all by itself was able to do this. We've replicated this now, not only ourselves, but other labs in multiple models for social dysfunction related to autism. So it's super exciting. We're trying to figure out mechanistically still, well, why. We have some inroads, but a really interesting finding that we have also is that it's not just any lactobacillus that can do this. Lactobacillus johnsoni, for example, had no effect on the maternal high-fat diet offspring behavior, right? So there's something specific that L. reuteri is doing to the host to improve social function in these tests. And so now we began to go into the brain of the maternal high-fat diet offspring, and specifically we looked for oxytocin levels in the paraventricular nucleus of the hypothalamus. Of course, all oxytocin is produced in the hypothalamus. Primarily, the paraventricular nucleus is central projecting. There's also actually some central projections from the supraoptic nucleus, but of course, most of the supraoptic nucleus oxytocin goes out to the periphery via the pituitary. So what we found was that there was this reduced level, kind of rostral to caudal, of oxytocin in the PVN that was then restored by rotary treatment. And so now we began to think about, okay, mechanistically, at the circuit level, what could be going on here? And we know that there's oxytocinergic output that goes from the paraventricular nucleus onto the mesolimbic dopaminergic reward system. And of course, social interaction with others, as you know, can be very rewarding, and that's what drives people to go back and seek additional social interactions. And there's of course output from these VTA dopaminergic neurons onto the nucleus accumbens, which has been shown to be a critical loci for normal social behavior. Now, importantly, this circuit is conserved between rodents and humans. So you can see a nice just illustration of that in the human brain. You've got oxytocinergic output onto the paraventricular nucleus, from the paraventricular nucleus onto the ventral tegmental area, which then sends dopamine to the nucleus accumbens, thus providing a salience to the importance of social interaction. So we wondered if there could be some sort of like physiological correlate of social behavior with a stranger, just like long-term potentiation in the hippocampus is a physiological correlate of learning and memory. Recent work at the time of Carl Dijsselroth's lab has shown that when they express channel rhodopsin, so this cation passing channel in the ventral tegmental area dopaminergic neurons, and shine blue light, they could stimulate these neurons. And importantly, in a behavioral paradigm, in that reciprocal social interaction test, they observed a significant increase in the amount of interaction that two male mice spend interacting with each other when channel rhodopsin and VTA dopaminergic neurons is being stimulated. In contrast, when they express halorhodopsin, which of course passes anions into the cell, thus silencing neuronal output, they drove a decrease in the total amount of time these two mice spent interacting. And importantly, this is specific to social behavior, as they saw no effects of either stimulation of channel rhodopsin or halorhodopsin on an object interaction test. So being familiar with this data, we decided to change up our reciprocal social task a little bit. And so in the experiments I'm about to show you, we not only did a stranger task with some of our mice, where the mice were coming from two different home cages. Now we also added a familiar interaction. And so these are two mice coming from the same home cage. And what we did was, this was a 10-minute test, so reciprocal social interaction for 10 minutes. 24 hours later, we isolated the brain, took transverse slices that included the VTA, and we recorded the AMPA to NMDA ratio. So this classic measure of plasticity in the VTA dopaminergic neurons, like in response to even addictive drugs like cocaine or amphetamines. And what we could see is, with a familiar interaction, there was no change in the potentiation of this AMPA to NMDA ratio. But at 24 hours following a stranger interaction, we see a significant increase in the AMPA to NMDA ratio that is on par with what you would see in mice that had taken cocaine 24 hours later. So importantly, this is also consistent with this very significant increase in interaction time with a stranger mouse versus a familiar mouse. And in our maternal high-fat diet mice, we saw no such potentiation following interaction with a stranger. So these mice were not interpreting social interaction as rewarding. They weren't getting that dopamine spike out of the ventral tegmental area. And we think that there's this downregulation of oxytocinergic output onto VTA dopaminergic neurons in our maternal high-fat diet mice. And consistent with that idea, they have an insignificant, very small increase in interaction time with a stranger over a familiar mouse. So again, the maternal high-fat diet mice have this physiological impairment that reduces social interaction in the animals. Now we're able to rescue not only the behavior of the maternal high-fat diet offspring treated with Lomasi lactobacillus reuteri, but actually we saw a significant increase in this potentiation of the AMPA to NMDA ratio in VTA dopaminergic neurons that were reported from in the brains of the reuteri treated animals. So we're able to restore this underlying physiological deficit with Lomasi lactobacillus reuteri alone. Now, so one question is, so is social interaction in this circuit from the oxytocinergic output onto the VTA? Is it dependent on activation of dopaminergic neurons there, or is it maybe inhibition of local interneurons? And so Rob Malenka's lab got at this, and they were using that social condition place preference test I talked about earlier with bedding A versus bedding B. And what you can see here is when they use oxytocin receptor flox mice crossed with DAT-CRE, so dopaminergic neurons free. So you see a clear preference developed in the control mice. So these are CRE negative, so these are control mice. But when you express CRE and thus delete oxytocin receptor expression in dopaminergic neurons, you lose that establishment of preference. In contrast, in the GAD2 CRE mice, which would knock out oxytocin receptor expression and VTA GABAergic interneurons, there's no loss of preference in the transgenic mice. And so this suggested that it really is oxytocinergic acting on those dopaminergic neurons in the VTA that's critical for social plasticity and normal social behavior. One question that we had at the end of the study was whether or not we could kind of bypass that gut-brain connection by just introducing oxytocin intranasally. There have been a lot of excitement recently about the potential for intranasal dosing of oxytocin to increase social behavior, even in humans. There have been some studies there, a lot of them out of Emory. And so what we did was we measured the AMPA to NMDA ratio in VTA dopaminergic neurons from maternal high-fat diet mice that were treated with intranasal oxytocin 30 minutes prior. So there's no interaction here. So anyway, oxytocin 24 hours prior to recording, there was no potentiation here. As we know from previous studies, there's no potentiation following vehicle interaction, a vehicle plus a stranger interaction. But when we did a intranasal oxytocin 30 minutes prior to interaction with the stranger, even in our maternal high-fat diet mice, this could drive potentiation of the AMPA and MDA response. It also rescued the reciprocal social interaction phenotype, as well as intranasal oxytocin rescued 3-chamber. And so that circuit is functional, is there, but there's some important part of the microbiota that's stimulating this oxytocinergic output onto these dopaminergic reward centers. And so after this project, there were several really interesting questions remaining. The first was, couldn't L-reuteri rescue social dysfunction in idiopathic and or genetic models for ASD, or is it really like a one-trick pony? Does it only work in the context of maternal high-fat diet where it's negatively selected by that maternal high-fat diet? And finally, of course, what is the underlying mechanism? And we began to address that in a Neuron paper that the first author on this one is Martina Scrita. She was a fellow postdoc in the lab, in Maro's lab at the time. And what Martina did was she used the Shank3B knockout mice. And so these are mice that have been used extensively by Jackie Crawley and others, great mouse behaviorists. And we actually asked Jackie at the time when we were developing the study, hey, what do you think is the best mouse with the most reliable social deficit phenotype? And she said it was the Shank3B knockouts. And so what you can see here is there's baseline deficits in sociability in these Shank3B knockouts that indeed can be rescued by this basically 4 weeks treatment with L-reuteri from weaning to 7 weeks, at which point we were doing the behavior. We see that not only in the 3-chamber test, but a clear rescue in our reciprocal social interaction test. Furthermore, we also observed the deficits in social interaction and induced synaptic plasticity. As you can see here in this pink, there's no significant potentiation of the VTA dopaminergic response following interaction with a stranger mouse, but this was able to be restored not only with L-reuteri, but also with intranasal oxytocin. And also you can see the effect of this circuit is obviously preserved when we applied cocaine, gave cocaine to the animals. 24 hours later, you see the strong potentiation of that AMPA to NMDA response. Not only did we show that worked in in a genetic model for autism, we found that it also worked in these BTBR mice, super cute mice. And they've got these yellow tummies and black back. But anyway, they have idiopathic autism. We don't know why these mice are socially dysfunctional, actually, but they just have baseline social deficits that could be rescued with Lomacki lactobacillus treatment. Furthermore, when we performed a monocolonization of mice that had been that were kept in germ-free isolators, that was also sufficient to rescue social behavior. And mice that normally see this white, these are baseline germ-free mice, so mice that came directly out of the isolator, but mice that were monocolonized with L-reuteri showed normal social behavior. So we wanted to know which which pathway, like gut to brain communication pathway, was this being mediated by? So one of the first things that we did was we performed subdiaphragmatic bilateral vagotomies. And what you can see here is in mice that were vagotomized and given L-rotary, that it actually attenuated the rescue. So that showed that the L-rotary rescue was dependent on vagus nerve integrity. And these are Shank 3B mice that have been given oxytocin. So even in the absence of that gut to brain communication, if you just introduce oxytocin intranasally, the mice show normal social behavior. Same thing in the reciprocal social interaction test. The next thing we did was we wanted to know what the role was of these dopaminergic oxytocin receptors. And so what we did was we created these mice, just like from the Malenka paper, we used DAC-Cree crossed them with oxytocin receptor floxed mice. And so in our controls in the black here, we've totally normal mice, no Cree expression. But in the mice that express Cree, we've got deletion of oxytocin receptors in all dopaminergic neurons in the brain. So this is a reciprocal social interaction data in C. But you can see normally, these are control mice, but when we delete oxytocin receptor expression, that actually impairs normal social behavior in those animals, and it cannot be rescued with either L-rotary treatment or even intranasal oxytocin, right? And so even though the oxytocin there, if it's not able to bind the oxytocin receptors on these VTA dopaminergic neurons and change their activity, that we don't get that normal social behavior, and also we don't get that normal social interaction and induced synaptic plasticity, right? So you can see across the board here, no change in the potentiation of the AMPA to NMDA response, but cocaine is still able to elicit a potentiation response because, of course, it's independent of oxytocin receptor expression. Following this study, actually, and what I'm going to do is I'm going to actually take you back in time, because this is a study that I actually started before all the Shank 3B work that Martina got started on. Again, we wanted to know, okay, so even models beyond Shank 3B, can it work in other autism-like autism models? And one of these that we worked in now is the catnap2 knockouts. And finally, is the L-rotary rescue exclusively mediated by the vagus, or could it be working via multiple channels? Yes, the vagus nerve is required, but there's other elements that are critically involved as well. So the work I'm going to tell you about in this section of the talk actually came out this summer, or actually maybe in April. So it was my final work out of Morrow's lab. And we found this paper totally went off somewhere that I didn't expect. So what we decided to do was use these catnap2 knockouts, which is a classic genetic model for autism spectrum disorder. It was really, this use was pioneered by Dan Geschwin's lab at UCLA. It was all based on some large GWAS studies that there was an association between the catnap2 gene and human autism. And so they made these catnap2 knockout mice. They have clear social deficits as well as hyperactivity. And importantly, why I wanted to start our experiments into genetic models for autism is because these catnap2 knockouts have been shown to have an oxytocin deficiency. And so you could rescue, the Geschwin's lab showed that you could rescue the social deficits with oxytocin. And interestingly, previously in a Cell paper from 2011, he had shown that risperidone, so that's actually an FDA-approved drug for autism, was able to rescue the hyperactivity phenotypes in these catnap2 knockouts, but had no impact on social behavior. But conversely, oxytocin rescued the social behavior phenotypes, but had no impact on hyperactivity, and that's going to get really important in a minute. So what I did was I ordered mice from Jackson. I just wanted to make sure that we would see, due diligence, see the phenotype, and we had beautiful social deficits and these catnap2 knockouts that are going to be shown in green throughout this work. And so that was all good. We also saw the hyperactivity report, that was reported as well. And so I decided now I was going to cross the catnap2 wild type and knock out like the recommended wild type lines, and then the knockout line to generate litters that had all three genotypes, right? So wild type, hets, and knockouts in the same litter, because that's the right way to do science, even though Jackson keeps these catnap2 knockouts on a homozygous knockout line. Much to my surprise, I found that when I crossed these animals to generate all three genotypes, there was a beautiful rescue of the catnap2 knockout social phenotypes. And so we saw totally normal social behavior in knockouts that were generated by this heterozygous breeding scheme. We saw this not only in 3-chamber, we saw it in reciprocal social, but we did see that the hyperactivity phenotype was maintained in the animals. So of course we began to wonder, okay, could this be actually driven by the microbiome? One of the first tests we did was to perform the co-housing. So we actually returned to our isolated lines of wild type animals versus the knockout homozygous line. And we performed co-housing at, from weaning. And then we waited 4 weeks, and then we did our behavioral analyses and collected fecal samples. And so what you can see was that there was a beautiful rescue of the knockout phenotype in the co-housed isolated line knockouts. And so this is true in 3-chamber, and then the showing of no significance in this reciprocal social test, of course, indicates that there was a rescue of the behavior. But again, we saw that there was no change in this locomotor activities. These animals still had hyperactivity despite being co-housed. Now, in 2016, Harold Virgin and John Stappenbeck from WashU actually put out a really nice perspective piece in Nature, where they were talking about how, as a community, we really need to consider how there's this combination of host genetics and microbial genetics that can create an overall phenotypic picture, not only in model organisms, but also in humans, right? And so this idea was really interesting to us. And so I'm kind of going to walk you through the logic here and see how this was clearly related to the phenotypes that we were seeing. So we have our control line and our isolated mutant line, our isolated knockouts, right? Read on a homozygous background. And we see this phenotypic difference. We see impaired social behavior in our mutant line, but normal social behavior, of course, in our controls. And now when we bred the litter mates and we test for phenotypic differences, what we saw was for social behavior, the mutants were no longer, for hyperactivity, the mutants were not equal to the controls. The mutants had a clear hyperactivity phenotype, but for social behavior, the mutants were equal to controls. We had a rescue of the social behavior. And so what this told us was that we've got, basically we hypothesized that the hyperactivity phenotype in the catnap2 knockouts was driven by host chromosome dominance, but that there was microbial genetics that were contributing to the social dysfunction in the catnap2 knockouts. And so now we began to test this hypothesis. So we performed 16S ribosomal RNA gene sequencing, again, saw a clear difference in the gut microbiome composition of the mouse lines bred separately, as you would expect, but no significant differences in the mice that were bred by heterozygous scheme. So we decided to perform fecal microbiota transplants either from the control line or the knockout homozygous bred line versus wild type and knockout donors from a heterozygous breeding scheme. Now, this is a lot of data. I'm going to walk you guys through it kind of pair by pair of columns here. So in conventionally colonized mice, we see a preference for mouse 1 over the empty cup, as well as mouse 2 over mouse 1 in the social novelty test. Now, baseline germ-free mice, so these are mice that are just immediately exited out of the germ-free isolator and put into our 3-chamber test. We see baseline deficits, so no preference for the mouse and no preference for mouse 2 over mouse 1 in social novelty. When germ-free mice are colonized with fecal microbiota from wild-type mice upon weaning, so this is at 4 weeks when they're exited from the germ-free isolators, at 4 weeks later, we see beautiful, social preference, both in sociability and social novelty. However, when the mice were transplanted with fecal microbiota from this homozygous generated catnap2 knockouts, we saw no rescue of the germ-free baseline behavior. However, in both wild type and knockout litter mates, when they were used as the donors for these germ-free fecal microbiota transplants, we saw that these both were able to rescue the social deficits. So this is consistent with the behavioral phenotypes, the social phenotypes that we saw in the 2 different lines, and there were no effects on locomotor activity. So again, we think this is really being driven by microbial genetics. So now we return to the question of whether or not a wordery could rescue the social dysfunction that we observed in the homozygous generated catnap2 knockouts. And indeed, we found that it did. So 4 weeks of rotary treatment rescued sociability, social novelty, as well as reciprocal social interaction in the catnap2 knockouts, but it had zero effect on the locomotor activity, so this hyperactivity phenotype in our catnap2 knockouts. And so this is consistent with the idea that oxytocin rescues social behavior in these animals, and we were able to reproduce that data from Dan Geschwindt's lab, but it has no impact on locomotor behavior. Furthermore, we found, like I said, we saw a clear rescue of reciprocal social interaction in our knockout isolated line, and also a rescue of the social interaction induced synaptic plasticity and VTA dopaminergic neurons. So we wanted to get more at, like, mechanistically, how is L-rotary working in these kind of 2 knockouts? And we decided to perform fecal metabolomics. So we did this in partnership with a company called Metabolome. And we saw it had several hits, the top 2 of which were biopterin and dihydrobiopterin. Now, these metabolites are in the tetrahydrobiopterin synthesis pathway. And what you can see here is we saw about a 20-fold decrease in the levels of both biopterin and dihydrobiopterin. That was almost completely rescued by treatment with L-rotary. So what is tetrahydrobiopterin? Well, it's an essential cofactor in the synthesis of monoamines, including even dopamine and serotonin. And so we think that it's basically feeding into this baseline deficits in this hypothalamic to mesolimbic dopaminergic reward circuitry that's so important for normal social behavior. So we decided to see whether or not tetrahydrobiopterin itself could actually rescue social behavior in the catnap2 knockouts. And just like L-rotary, we see that it does rescue social behavior in the animals, but again, has no effect on hyperactivity, just like we saw with the L-rotary rescue. It was also able to rescue social interaction due to synaptic plasticity in the VTA dopaminergic neurons of the homozygous knockouts. Importantly, we also found that we were able to, when we blocked the enzyme in the final synthesis pathway of, which is called of tetrahydrobiopterin, called sepiapterin reductase 3, we were able to attenuate the rescue of L-rotary. So it does seem to be essential, the tetrahydrobiopterin pathway and the effect of L-rotary on this does seem to be absolutely essential to normal social behavior. And another really great finding that we had was that blocking that sepiapterin reductase 3, so blocking the synthesis of tetrahydrobiopterin, was sufficient to block normal social behavior in even just wild type mice. And what's interesting about this is, like, there's another drug that I don't think it's FDA approved yet, but called Kuvan, that is tetrahydrobiopterin, that has some interest in being an autism treatment for social dysfunction. So that's really exciting. And it's neat to see how these microbiome studies can identify metabolomic targets that could potentially be used in therapeutic applications. So yeah, so just to summarize this work, we find that the host and microbial genetics are contributing to overall phenotype in these catnap2 knockouts. Now there's several studies even this year that we've seen in review and also that are beginning to come out, showing this combination phenotype. So some host genetics and microbial genetics generating an overall phenotypic picture. And I think that we're going to see a lot more of that as time goes on. So one question that we've been asking in my own lab now is whether it's the existing state of that adolescent gut microbiome in our maternal high-fat diet mice. So this is all returning to that maternal high-fat diet that really is causal in the social dysfunction, or whether high-fat diet induced dysbiosis in the maternal gut microbiome could impair early life neurodevelopment. And so we presented this work recently at the Society for Neuroscience meeting. And what we know from my early work as a postdoc is that we've got in males, we have dysbiosis of the gut microbiome and social dysfunction. However, of course, given the vertical transmission of gut microbiota from mom to offspring, we also hypothesized that the female gut microbiome would be dysbiotic as well. And we wondered if this dysbiotic female gut microbiome, given the endurance, that long-lasting dysbiosis of the gut microbiome, I showed you the data in that principal coordinates analysis, like 6 months on, right, that showed clear dysbiosis, whether this could lead to secondary effects through behavioral dysfunction in the F2 males, despite these F1 females being fed a regular diet. And so what we did was, this is our breeding scheme. So the only animals exposed to high-fat diet were the grandmaternal mice. And now we're focusing really on this F2 generation. Despite a regular diet throughout, after weaning and mom in this generation, whether or not she's still got if she maintains a dysbiotic gut microbiome, and the long-term impacts on the F2 offspring. And what you can see here, this is actually 16S ribosomal RNA gene sequencing of the F1 female gut microbiome. And just like in the males, we've got a significant reduction in overall alpha diversity, right? And we've got a clear distinction as principal coordinates analysis. But very interestingly, in our F2 animals, we're actually beginning to see kind of a partial rescue of the F2 maternal high-fat diet descendants. And you can see that here in this observed OTUs graph. And so there's an increase in the overall diversity, right? But there's still this distinct beta diversity. We've got several hits here that are either they're decreased or increased in the maternal high-fat diet F2 descendant microbiome that we're looking into mechanistically. How could they be contributing to phenotypes that we see in these animals? But speaking of phenotypes, even though their mom is fed a regular diet from the time of weaning, we still see this profound social dysfunction in male F2 offspring. So you can see that here. And this data is perfectly matched. This is the observer data from interaction, but we also see this perfectly for the automated time and chamber data. We also showed that elroidery is able to rescue the deficits in our maternal high-fat diet F2 descendants. And so this is really beginning to kind of change the way that I think about elroidery. And I think that elroidery is just kind of working to fix this overall dysfunctional system in the oxytocinergic to hypothalamus to mesocorticolimbic dopaminergic reward system, no matter in which mouse model we're talking about, right? And so this is all throwing the focus back on mom's gut microbiome. And so we also perform fecal microbiota transplants from F2 donors, either maternal regular diet males or maternal high-fat diet F2 males into male germ-free recipients, waited 5 weeks, assay behavior. And interestingly, in contrast to what we saw with the F1 transplants, we saw that both maternal regular diet F2 gut microbiome, as well as the maternal high-fat diet F2 gut microbiome, was able to rescue behavior. So again, this throws into question whether or not that male gut microbiome is really causal in the social dysfunction, or whether there's effects going on during fetal neurodevelopment that are critical. And so what we're thinking about now is that we've got this high-fat diet, which leads to dysbiosis of the maternal gut microbiome and adverse development in the F1 animals that leads to brain and behavioral dysfunction. And that the same thing is likely happening in the dysbiotic gut microbiome of F1 females, leading to brain and behavioral dysfunction in their male offspring. So in concluding perspective, this is a picture of Mabry Mill Road maternity hospital in Liverpool from the 1960s. There was a great deal of really important work done with folic acid supplementation in this relatively poor population that had a high incidence of fetal malformations, including neural tube defects. And so a doctor here began doing these kind of large-scale trials of treatment with folic acid. And as you all know, folic acid is now almost universally used as in women during pregnancy and even before pregnancy to prevent these neural tube defects. And it's remarkable how effective and cheap it is, right? And so now here's a picture of the modern facility here in the same site, which is now Liverpool Women's. And I think that we're at this exciting period where prenatal probiotics could be just as therapeutically meaningful today as prenatal vitamins that included folic acid supplementation were in the 1960s, because I think that we're at a point in this kind of hierarchy of maternal fetal care provisions, kind of this Maslow-esque hierarchy where we've dealt with perinatal mortality. We've really reduced that. We've been able to reduce fetal malformations by use of prenatal vitamins. Of course, the child mortality is down, especially in developed nations. And finally, I think we have this privilege of being able to focus on mental health of babies. And I think that prenatal probiotics, basically just to normalize the maternal gut microbiome, to provide some anti-inflammatory or at least immune modulatory effect with commensal probiotics is really where we're at right now. And I'm excited about the therapeutic potential for these. So again, we're just super excited about how the promise of targeting the gut microbiome, it's so much easier to target the gut microbiome than it is to change human genetics, right? But as we've been talking about throughout this whole talk, it's this important chemical factory that generates these bioactive metabolites that are even neuroactive, right? And so we can do this through dietary means. We can change the microbial ecology of the gut through different probiotic and even strains and prebiotics. And this can alter the metabolite output of an individual's gut microbiome so as to normalize brain function and behavior in offspring. So I'd really like to thank the members of my own lab. This is Claudia and Lisa, Robert and Ian here at UTMB. We've got collaborators at Baylor that we work with for our metagenomic sequencing. So that's Christy Hoffman, and she works with Joe Petrosino. Of course, the work that I presented earlier was primarily performed in Morocco Samadioli's lab. I love doing my postdoc in that lab. Sean Dooling was a student that helped with the CatNap2 paper. Martina Scrita, of course, did all of the Shank 3B knockout work. Gonzalo Viana Di Prisco did all of the amphidam-MDA ratio, all the EFIS in our 2016 paper. And then these are team members that helped with all of the sequencing and data analysis for that. So thank you guys so much. Obviously, I'd like to thank our sponsors, several institutes within UTMB, the Gulf Coast Center for Precision Environmental Health, the National Institute for Mental Health funded some of the studies in Morrow's lab. And then, of course, my own lab's funded by the Brain and Behavior Research Foundation, as well as the Scott Gentle Foundation. Thank you so much.
Thank you, Shelley, for such an interesting and enlightening presentation. We have a number of questions in the chat. I'm going to read them, and Paula is monitoring the chat in the YouTube channel. So if somebody wants to ask a question and they want to ask the question live, please just raise a hand or just turn on your camera and microphone. So Eugenio Contreras asked, give me a second. Does this phenotype remains if cross-fostering after birth? And I guess that he's referring to the phenotype that you first talked about in your 2016.
Yeah, great question. We've actually not done the cross-fostering, so that's the question of, is it a gestational exposure or lactation? I think that's an awesome question. Certainly one we're interested in pursuing. I think that, obviously both periods are critical, especially neurodevelopment periods. And so I think teasing that out will be really valuable. It just all comes down to, time and money to be able to do the various experiments, but great question.
I think Gabriel has a question. Gabriel, do you want to ask the question?
Yeah. Hi.
Hi.
Very nice seminar.
Thank you.
I wrote in the chat that I was surprised to see how easily you can, the microbiome will accept a microorganism. But I don't remember if you actually measure the microbiome composition after you fed the mice with this microbe.
That's a great question. We didn't. We did a very small pilot test that was insufficiently powered, in my opinion, so I never showed that data. But we've actually done it in our F2 animals, and it does have a profound effect on the overall microbial community, especially in our maternal high-fat diet offspring. So we think that the dysbiotic state is more unstable, right? So it's going to have a larger impact on, some species, their abundance is increased, some is decreased. But importantly, and to your point in the chat here, we are dosing these mice every day with L-rotary, and they have to stay on L-rotary for us to see this effect. We've actually done experiments where if we take them off L-rotary, we begin to lose that social phenotype. So for example, if you take the mice, if you do an L-rotary treatment from weaning to 7 weeks of age, and then you take them off for the subsequent 4 weeks, and then you test behavior at 11 weeks, then we see normal sociability, but a loss of preference for social novelty, right? So we're beginning to see that loss of phenotype. Jim Versalovich's lab, he's at Texas Children's Hospital, they did a lot of looking into, so the strain we use is L-rotary 6475. It was isolated from human breast milk by a company called BioGaia. So it's a human strain that we're putting into the mouse gut. So it actually doesn't colonize. So if you look for it like 72 hours later, you won't find any significant increase in L. reuteri, but somehow, and this is a strain that does produce reuterin. And so that's actually an antibiotic. It will actually kill other strains that compete in the same niche. So yeah, it's a really interesting question how it's doing it, and the question of, and I think we're not there yet, like in probiotics. Are we delivering, like, what's the right mechanism? You know, can we actually get things to colonize, or will we need to be constantly taking, you know, probiotic pills? I think that those questions are still unanswered.
Okay, thank you.
Sure.
We have another question in the chat by Violeta. Are there any clinical trials with L. reuteri in patients?
There are, yes. I think there's one out of UT Southwestern, and then there's an Italian trial going on right now. I do not think the results are out yet.
Second question by Eugenio. New data indicates that maternal immune activation, for example, obesity, leads to epigenetic changes on T cells in the offspring, leading to impaired social behavior. Yep. In this case, the T cell itself can induce impaired social behavior. Have you tried to perform the HFP experiments on animals that lack T cells, like rat?
We haven't yet. That's certainly on our radar. We're really interested in this gut microbiota immune brain crosstalk, right? And so we're certainly interested in that angle, but we've not yet tried that. But I anticipate, you know, it's easy to have just kind of, oh, it's a single, you know, communication pathway. It's just the vagus, or it's just the metabolites, right? But I think that it's really this synergy of those of these pathways working together. That is how L. reuteri is able to affect an overall system and result in the behavioral rescue that we see. So certainly, I think there's immune elements playing out there.
Another question by Elena. It is clear that there is a sex dimorphism response in many events, including programming. Why only the analysis of male offspring? Do you have female data? Are they different? So yeah, so I agree. There's a lot of sexual dimorphism. We do not see the same level, or of social deficits in our females, in female offspring in either the F1 or F2 generation. Interestingly, females in the social novelty task of the 3-chamber test actually don't show a preference for mouse 2 over mouse 1. These are just even like wild type normal female mice. And I think it's important to remember that these behavioral tests that we use so frequently in assessing these mouse models for autism were designed to test male social behavior. So I think that we're really not getting to female specific social behavior. I'd really like to integrate more like tasks like Rob Fromke's developing in his lab up at NYU, that's more like alloparenting and some other very female specific social behaviors to really tease that out. So in our 3-chamber test, yeah, they don't have deficits, but maybe we're not looking at the right behaviors in females. Jasmine, do you want to go ahead and ask your question?
Yes, I have. Thank you. Hi, thank you for the talk. Very interesting.
Hi, sure.
I have, well, 2 questions. Quickly. In the first, did you measure instant to just AMPA or NMDA currents? Did you measure LTP, LTD? I mean, more.
We didn't. We measured H current just to make sure that we were in dopaminergic neurons, right? We were working in the ventrolateral VTA, but we didn't measure like field potential stuff. So we haven't looked at.
And that was in the VTA, right?
Yes, it was all exclusively in the VTA.
Okay, that gives me the other question. Since you don't see effect in the locomotion, I mean in the anxiety, do you think that the microbiome has effect in a specific circuits? What I mean is that maybe if you record in the basal ganglia, or I mean in the striatum, do you see the same effect?
Yeah, that's a great question. So we haven't recorded in the basal ganglia or the striatum of these mice, but I think you're totally right. One other set of data that makes me think of that is Elaine Tsau's paper from when she was working with Sarkis Mazmanian at Caltech. And so that's a 2013 paper in the maternal immune activation model for autism. And of course, she identified Bacteroides fragilis, was able to rescue some of the autism-like phenotypes in the maternal immune activation model. But importantly, so it rescued, for example, like marble bearing, which is of course like for perseverative behavior, and also so communication deficits, so the changes in USB, but it had no impact on the 3-chamber test, right? And so it does seem like these different bacteria might have, circuit specific effects. And, I think that it's very complicated, like the underlying mechanism by which they would do that. But it does seem like, an overall like cocktail approach would be better, right? Like, so I'm really interested in like, okay, if we combine, Bacteroides fragilis plus Lactobacillus reuteri, can we rescue both, these hyperactivity phenotypes as well as the social phenotypes we see?
And what about the grooming? Sorry, the last.
Yeah, we never quantified grooming, so I can't tell you anything about that. I didn't notice any like obsessive grooming, so just kind of qualitatively, but we never quantified it.
Okay, thank you so much.
Yep.
Diana asks, has L-reuteri or BH4 been used to treat depression?
I don't know. I don't think so. L-reuteri is a very common probiotic. I mean, you can buy it on Amazon, probably at a grocery store in downtown Mexico City, I'm sure. So it's been used for a lot. I think it really does have these amazing effects on the host. I'll have to look into that. That's not really my area of expertise, but I don't know.
I think there's a couple of questions from the YouTube channel. Paula, do you want to read them?
Sure. So let me just quickly. Moises Edith asks, have you identified the microbial metabolites from L-reuteri that might be responsible of the observed effect?
So that's something I know Mauro Cosmadioli's lab is working on. So it's something that we kind of wanted to keep our hands off, right? Just as we're establishing independence. So we haven't. One thing I can tell you is that when we introduce heat-killed L-reuteri, it doesn't have a rescue effect. So it does seem to be, we need to have the live bacteria to have the effect. And so we do think it's generating some critical metabolites. I can't tell you what those are right now.
Okay, and he has a second question, which is, according to your findings to this date, a priori, what will be your single recommendation to pregnant women in order to prevent this condition? Congratulations on your fantastic research.
Thanks. I would certainly say that, diet is really important, right? Like keeping that, the leafy green diet that'll, provide these complex carbohydrates to many commensals that thrive on them, as well as, salmon and other things that are high in these omega-3 versus, the more like inflammation-inducing omega-6 fatty acids. And so I think that the importance and what we saw in our maternal high-fat diet model is that, and we of course took it to the extreme, right? So the diet that we're putting these animals on is like 60% fat, but obviously that reduces, and the microbiota is extremely sensitive to the stoichiometry of macronutrients available in the diet, right? And so I think that a lot of these just the bacteria that thrive on complex carbohydrates that are available in leafy greens and other incredibly healthy food are just negatively, they're just pushed out by these poor diets, and they just aren't able to regain in abundance. And I think that a lot of those species are immunomodulatory, right? And so it's really helping to dampen down, hopefully, maternal inflammation, during pregnancy. So yeah, for moms, healthy diet. I'm not sure, on like, I can't recommend like a specific probiotic. I think we're en route, but I don't want to say anything right now about that.
Awesome. I just have a question related to this last question.
Sure.
Have you looked at the epidemiology data in humans? Like, what's the frequency of autist children from obese mothers, for example?
Yeah, so one of the best papers I saw on that is by Panjwani et al. From 2019. It was published in Autism Research, and it was a really nice paper showing that there was actually a 10-fold increase in the odds ratio for autism spectrum diagnosis in women that are of obese status, and also for male children, they had a specific metabolite that was also implicated there. And so I think there is this, anywhere in the literature from about 2.5 to 10-fold increase in incidence of a neurodevelopmental disorder in general in kids that are born to women of obese status.
Thank you. Very, very nice job. Thanks.
Thank you.
I have a question. Do you think dysbiosis could make addiction worse or improve it a little bit?
Yeah, that's a really good question. Absolutely. I think that there's some work that I know about that I don't think is published yet, suggesting that, addictive drugs even like work via, different microbiota and have different effects on, like their effects are dependent on gut microbiome composition. It's really fascinating. And so absolutely, I think in a lot of individuals that are addicted to drugs, so it could do two things, right? It could drive a predisposition toward addiction, or individuals that are addicted to drugs often, they seek those above anything else, right? And so they're not often, pursuing a healthy diet, right, or exercise or anything like that. And so, the gut microbiome in those individuals is also just going to be compromised by lifestyle, right? So I think it's certainly a contributing factor that could exacerbate, addiction in the underlying, neuropathology.
Well, thank you so much. I think that's all the questions that we had. And thank you everyone for joining, and happy holidays to everyone.
Yeah, happy holidays.
And Shelley, if you got two more minutes,
I'll stay for the students. Yeah,
she wanted
no
to ask
problem.
a couple of questions. Thank you everyone.
Yeah.
Feliz año nuevo.
Felices
como sea.
fiestas.
Felices fiestas.
Gracias, felicidades, gracias.
Thank you so much. Great work.
Felicidades.
Very
Que les vaya
nice
bien. Gracias.
seminar. Thank you.
Gracias.
Thank you.
Happy holidays to everyone.
Yeah,
Bye.
Merry Christmas.
Bye.
Thanks for attending.
Felices vacaciones.
Thank you, Shelley, for the extra time.
Oh yeah, you're welcome. No problem. I love this. We do this after our seminars and at UTMB as well. I think it's really valuable.
Yeah. So, Yvette, if you want to stop the transmission on the YouTube channel, that would be a good moment. Yvette, si quieres terminar la transmisión en YouTube, estaría bien. No sé si me yo. Bueno, si no creo que importe mucho. Sharini, ¿quieres empezar con las preguntas?
Thank you, Dr. Luis, and thank you, Dr. Shelley, for staying. I have a couple of questions since I'm with the communication unit of the Institute.
Okay, right, yep.
I'm doing a story on your work.
Oh, great,
So
thank you.
I thought it would be interesting that you would share, what made you interested in this type of research? What made you interested in autism or microbiota?
Yeah, great question. So when I started in Mauro's lab, I was really interested in doing more autism research. I love, like learning and memory, and then also like, intellectual disability research, everything I had learned in my neurobiology of disease classes, et cetera, when I was an early graduate student. But the graduate lab that I joined, I thought it was an excellent environment for learning some basics, cell biology of the nervous system. And I really liked the training environment there. So I thought I'd, basically develop my baseline, neuroscience techniques there. And then in my postdoc is where I could really focus with my interest in doing science, really focus on neurodevelopmental disorders and specifically autism. And so I got the privilege of being able to do so when Mauro invited me to join his lab. And we actually began looking at the effects directly of high-fat diet on animal behavior, and didn't really see any, profound phenotypes, even on, cognitive deficits. And so that was surprising to me because I thought it would have a strong effect, but we really didn't see it in that first generation, that direct feeding. And so at the time, that was about 2012, I heard this really interesting talk by Stacy Bobo at the Society for Neuroscience meeting in